Welcome everyone. Thanks for joining today. For anyone that's might be attending this call for the first time, we've been holding this industry-wide call every month with companies that want to contribute to the cultivated meat value chain. And so the purpose of these calls is really to offer new and existing cultivated meat stakeholders an opportunity to learn from each other with respect to obstacles as well as potential solutions that exist along the value chain, with the end goal of hoping to accelerate R&D and commercialization throughout the field. So if your group is interested in presenting on a future call, maybe you're working with a company that could benefit from attending these calls, please feel free to put them in touch with me directly and we'd be happy to get them added. So a couple of housekeeping items before we begin from my side. The first is on surveys. So thanks to everyone who filled out this quick 5-minute survey regarding interest in focus areas for the Cultivated Meat Consortium at Tufts University. If you haven't taken the chance and you might be interested in such a consortium, feel free to fill out that survey. It's just, a couple of minutes, couple of questions. I'll drop the link in the chat there. Secondly, we started another work stream recently on the techno-economic analysis of cultivated meat, and we're going to be sending out another sort of fairly lengthy questionnaire next week, where we're going to ask folks questions on cell parameters, kinetics, sensitivities to metabolites, nutrient consumption, those sorts of questions. Reason being is that we really need some real world data from cells that are being used in cultivated meat manufacturing to hopefully create more accurate and representative models. And so this is really one of the best ways that you can support the work that we are doing. So if you're a cultivated meat manufacturer or you're working with some of the relevant cells, please be on the lookout for this survey in your inboxes next week. And then finally, next week, I also want to highlight, we have another sort of sister seminar, the Business of Alt Protein. And on that call, we'll have the team from the Cultured Hub, which is a new pilot scale facility in Switzerland that is opening for cultivated meat companies. And so you might be interested in attending that and learning more about their sort of CDMO services in Europe. So I'll drop a link to that in the chat as well. And then finally, pleased to be welcoming James Sherley from Asymmetrex today, who's going to share more about challenges and new methods in quantifying stem cell populations in your cultures. And then we'll turn it over at the halfway point to Chris Munchinsky from InflexionPoint, who's going to discuss some of the challenges and solutions for traceability systems. So feel free to drop questions in the chat throughout, and we'll get to them at the end of each presentation. I will turn it over to you, James.
So thank you, Ellie, for that introduction. I also want to thank GFI for the opportunity to share some concepts and technologies that Asymmetrex is developing that we think could be helpful to the effort of producing cultured meat and other food cells. And so the topic of my talk today is looking at the bioengineering of tissue stem cells to arrive at more effective long-term natural food cell production. My contact information is here. It'll also be at the end of my talk. So to get started, here's the outline for the talk today. I want to first talk about some principles of vertebrate tissue cell kinetics in the bodies of vertebrate animals that actually translates to how well we can culture their cells for tissue cell production. I want to introduce you to a new tool, differential tissue stem cell counting, developed at Asymmetrex, that we think can help to improve our ability to have better production of natural tissue stem cells for cultured food production. And I won't have time to tell you much about how that new tool works, but you can find out about that on a page on our website called How We Count. And then finally, I want to give you some examples of how you can use differential stem cell counting to improve bioengineering of more effective and more efficient production of cultured natural food cells. So this is sort of how I think about what I've learned about this field, is that the idea is that we have these farm animals, like beef and pork, and we want to move from making more of these animals to taking cells from them and doing industrial scale production of these cells for food products, ideally in big reactor systems like this in the future. So towards that goal, I want to really, my main focus today is to sort of get people thinking about the fact that understanding how vertebrate tissue stem cells work in the bodies of animals is important to being able to culture natural tissue tissue cells for food. And I want to begin by this question for us today, which is, why don't vertebrates grow indefinitely? This is the answer to this question underlines everything else I'm going to say. All right, so we know that things like cows and pigs and chickens and even fish, they don't keep getting bigger and bigger and bigger. They grow to a fixed body size, and then they don't get bigger than that. And the reason for that is that vertebrate tissue cell physiology is based on the asymmetric self-renewal of tissue stem cells. And that's shown here. All of the tissues in vertebrate bodies have tissue stem cells that are a small fraction of the total cells. They undergo this very specialized process called asymmetric self-renewal, which means they divide and maintain their own stem cell state while giving rise to cells that then produce the differentiated lineage of the tissue. So there are initially these committed progenitor cells that have a finite number of divisions, and they give rise to the differentiated cells, which we're interested in for meat and for food. And I just want to give some examples of this is throughout the tissues of vertebrates. And some well-known examples are the hematopoietic system, where they're asymmetrically self-renewing hematopoietic stem cells that give rise to cells that differentiate. And then they form the differentiated cells of the blood system. Two other examples, in liver, there are this area called the portal triad, where there are biliary epithelial stem cells and hepatocyte stem cells divide asymmetrically, maintaining their own state, continuously giving rise to the biliary epithelium and hepatocytes for liver function. And one of the best known examples is the villus crypt unit in the small intestines of mammals, where there are stem cells that divide asymmetrically in the crypts. They maintain their own state while giving rise continuously to cells that form the functional villus of the intestine. So this is throughout tissues. And so if these things are going on in tissues, they probably matter. And I want to talk about how they do matter when you're trying to grow these tissues in bioreactors. And so the first principle I want to share with you is that these in vivo tissue stem cell kinetics continue in cell culture. And that's based on 20 years of research at the Fox Chase Cancer Center, Massachusetts Institute of Technology, and the Boston Biomedical Research Institute that underpin all of the technologies that are being developed at Asymmetrics. And so the basic idea is that in culture, tissue stem cells can divide, and they primarily divide asymmetrically, giving rise to cells that differentiate from committed progenitors to these terminally arrested cells. In the body, these cells turn into the mature tissues in the organ or in the tissue. In vitro, they just simply undergo a terminal arrest. And we spend a lot of time in trying to now get these differentiated cells that are arrested to make the foodstuff products that we're interested in using for human food. It's important to note that stem cells also have the ability to divide symmetrically. They do this rarely in tissues. It's a very important process when animals are growing and getting larger to make more tissue units. It's an important process for repairing tissues when they're damaged to make new tissue units. But it's highly regulated because, as you can imagine, if this is out of control, then you end up producing large masses of stem-like tissues, which are tumors. And one of the things that underpins Asymmetric's work is our early learning that a very important cancer gene, the p53 gene, in fact, regulates this decision by stem cells, whether they divide symmetrically or asymmetrically. Now, the second principle, which is also very important to understand, is that because these kinetics continue in cell culture, they limit our ability to produce natural tissue cells in culture. I want to go through the explanation for that concept here. So if you think about this as a primary tissue isolate, where there are going to be some rare, less frequent tissue stem cells shown by these bivalent cells here, There'll be some committed progenitor cells in that initial preparation, and there'll be some differentiated cells. So if we look at the tissue stem cell fraction in the starting example here, it's about 1 in 5. Now, if we put this into a reactor and let this undergo, in this example, 4 generations of growth, these stem cells, they regain the production of what are called these turnover units here of committed progenitor cells. And in culture, these cells, although they may not fully differentiate, they arrest. So this culture will just simply start accumulating these terminally arrested cells. And at the end of 4 generations, the stem cell fraction now is about 1 in 30 because of the asymmetric division, which does not increase the stem cell number. Now, if we take this reactor and try to make more reactors by dividing these cells, and let's say we do a 1/3 reactor split here, the new reactors will get roughly this population of cells here with fewer total stem cells. But those stem cells will still give rise to the committed progenitors, which will accumulate again these differentiated arrested cells. And now the stem cell fraction in the new bioreactors is 1 in 64. If we continue this process to do more and more expansion, we eventually start reaching bioreactors that have no stem cells. And since the stem cell is the only cell in the system that can divide indefinitely without a lineage arrest, these systems will eventually reach a population of completely arrested cells. And this is the fundamental problem of trying to make mature cells in culture, the dichotomy between a cell that produces them that divides asymmetrically. So now I want to talk about how do we translate these principles into solutions for making more of the cells that we want, these differentiated tasty cells here for meat, in culture when we have a system that undergoes these asymmetric divisions as a primary default. And the answer to that is simple. We need to have more of these symmetric divisions, which are limited in the body. And what we've shown in our research earlier is that they're also limited in cell culture. So the solution here is to figure out a way that gives rise to these differentiated cells, which we want for beef and pork and chicken. Now, there have been some approaches made at this attempting this using induced pluripotent cells. And the problem with those cells is they actually don't know how to do this naturally, and we have to train them. There's a lot of work going on trying to get them to differentiate. Also, in our probability, induced pluripotent stem cells may very well go through this type of intermediate in order to produce the downstream differentiated cells. So in order to bioengineer this process to achieve this type of balance between producing stem cells and having them producing the differentiated cells, we need tools. We need tools to be able to quantify the number of stem cells in reactors. We need tools to monitor changes in their fraction. And we need tools to figure out how to obtain this type of process and to be able to control it. And so we have developed such a tool, and that tool is called differential tissue stem cell counting, and I want to tell you about that now. So there's been a long-standing problem with tissue stem cells and being able to quantify them. We call it the tissue stem cell counting problem. And this slide illustrates what the problem is. So currently, there are no stem cell biomarkers that are specific for stem cells. The current biomarkers that allow you to quantify these cells are actually stem progenitor biomarkers. They're expressed on the surfaces of committed progenitors as well as on the surfaces of the stem cells. And so when you do a count with one of these biomarkers, you actually count both. And you can't follow accurately how many stem cells are present, and you certainly can't follow what they're doing. Now, there's one exception to that statement. There is a biomarker that is a nuclear transcription factor for satellite stem cells and skeletal muscle. It's called PAX7. And PAX7 can, in fact, be used to follow and monitor these types of cells. But it's not very convenient because it's a nuclear marker, not a surface marker, which would be much better for doing research and method analysis. So our differential stem cell counting method allows you to quantify just the stem cells. So this procedure is called kinetic stem cell counting that allows us to do the differential stem cell count. And to be more specific, what our kinetic stem cell counting process does, it allows you for a complicated, heterogeneous culture that contains each of these 3 cell kinetic types to delineate the stem cells from the committed progenitor cells from the terminally differentiated cells. And to give you an example of what we can do with kinetic stem cell counting. This is sort of a profile for the expansion of cells in culture. We're looking at the number of cumulative population doublings over time. So we're looking at the expansion. And what's typical for all vertebrate cultures so far, if you don't do anything else to them but just culture them, they all eventually reach an arrest where you don't have any more doublings. What our kinetic stem cell counting process allows you to do is to follow independently the behavior of the stem cells, which is shown here. You can see, as I told you earlier, there is this decrease in the stem cell fraction, a rapid decline in time. To follow the behavior of the committed progenitor cells, shown here in red, and the production of the terminally arrested cells. And what you see from our analysis is, in terms of quantitative analysis, is what we know occurs biologically, because at the end of this culture here, there are no dividing cells. And that is the case here as well in this analysis. So we're able to delineate these three different kinetic compartments over time. And that then allows us to have some very unique applications that we think can benefit bioengineering, more effective and more efficient production of cultured natural food cells. And I want to just give you a few examples. So one of the things that we can do with this method is to count stem cells in a sample, which wasn't possible before. And we're able to do this with what are called rapid stem cell counting algorithms. So if you have one of these algorithms, and we've derived many of them now, you can take a bioreactor, take a sample from it, culture that sample for 72 hours, just do a total general cell count. If you put that information into our algorithm, at any day in the expansion, that data is used to get the population doubling time. These algorithms convert that into the stem cell specific fraction. So you can, in fact, go to our website and try some of these calculators now, because we offer them to the community for free for analysis at our Stem Cell Counting Center page. The ones that are there now, because our work has started in trying to produce human cell populations for human therapies, they're all for human tissue stem cell types, but we are currently developing them for animal stem cell types and tissue types as well. Those will be forthcoming. So another example of what we can do by being able to quantify stem cells differentially is to predict how well a culture is going to produce in the future. And this is a problem, certainly we have seen it in the human therapy industry, where you have a starting sample of cells, you want to make more of those cells. And what you know is that the production is variable. Sometimes you get poor expansion, sometimes you get good expansion. So trying to figure out whether that's going to happen is important, because it costs money to have an expansion that in fact is not successful. So what I'm showing you here is that we can do a differential count on these mesenchymal stem cell populations here. And we can measure how the stem cell fraction changes during the first 3 days of the culture of a sample. So if there's no change, it's 0 on the scale. And as I've already shown you, in most cases, the stem cell fraction decreases with time. So most of these samples that we looked at here for this series, they're actually showing that, in this case, this would be a 50% decrease in the stem cell fraction during the first 3 days. By looking at that measure, we can predict whether a culture is going to expand poorly or expand at a high rate. And we do this with a reasonably high degree of confidence. So we think this is going to be a very important tool for developing bioreactor processes and for quality control. So you can know at the beginning whether or not your sample is going to grow well in the future. And this is actually looking at samples grown out as far as 4 weeks later. And then lastly, what I want to share with you is that using our kinetic stem cell counting method and differential stem cell counting, we can actually learn why cultures do well and why they do poorly. And we can identify factors that govern whether that happens. So this is an example here where we're showing the same starting preparation of these. I think these are mesenchymal cells, mesenchymal stem cell populations, and growing them with different growth factor supplements. So this is controlled. And in fact, this is Thermo Fisher's supplement for mesenchymal stem cell production. This is fetal bovine serum, commonly used in research. And then these are 2 different suppliers' human serum as a supplement. And you see right away that in this expansion, these growth factors do a lot better for maintaining the growth of these cultures over time than the human serum. With our kinetic stem cell counting, we can probe the underlying cell kinetics and then understand why we see this difference. So shown here is the kinetic stem cell counting analyses. And we're just showing that paradigm that I showed you earlier, where there are stem cells that can divide symmetrically or asymmetrically. And it becomes readily apparent why there's this difference in expansion. The first thing is that in these human serum cases, the stem cell death rate is about 50%. Half of the stem cells produced in the system are dying. All right. Up here, you can see the death rate is much lower for these other supplements. Another big factor, which I showed you earlier, we've sort of achieved here with these growth factors, is that the symmetric rate of stem cell divisions is more than 50%. So we have a pool of cells that can revert to producing the cells that we're interested in here, if we were talking about making food cells or differentiated cells, right? And that's also true in both cases. You can see here it's about 61% symmetric divisions. While in these cases here, those symmetric divisions cannot sustain the stem cells because they're too low at 18 and 16%. And finally, another feature that we can now understand too is how stem cell dilution takes place in these cultures. You'll note that these turnover units here, the total number of cells predicted to be produced after 10 to 16 divisions is about 130,000 cells that are going to dilute the stem cells that are there producing them. In this case here, it's about 33,000. You note these turnover units are smaller. This is about 256 cells, so they're not going to dilute the stem cells that are dividing symmetrically nearly as rapidly. And the same thing here, there are about 8,000 cells produced in these units. They're not going to dilute these stem cells that are dividing symmetrically. So it's that balance that governs how many total cells we get to make. And I haven't shown you here, but they're on the order of 100 times more cells produced here than what are produced there. So I just want to close with taking you back where we started. We think that it's very important to apply these principles of understanding how vertebrate tissue stem cells grow in the bodies of animals to how we're trying to produce them when we're trying to make more of the natural stem cells, tissue cells in culture reactions. I've introduced you to differential tissue stem cell counting, which we think is going to be a powerful tool, not only in what we're doing in cell therapy, the parallel cell production universe to cultured meat, but in cultured meat to help improve the processes by which we get long-term production for cultured food production. And then finally, I've given you some examples, some early examples of how this can be applied. And I think that more application of this is going to be beneficial to this industry. And we're excited about participating in that. So I want to thank you for your attention. If you need additional information, you can contact me directly. Also, you can just go to our website, asymmetrex.com, and learn more about what I've told you today. So I'm going to stop there. I'm going to stop sharing my screen, and I look forward to questions and comments.
Awesome. Thank you, James, for that very clear presentation. If folks have questions, feel free to come off mute or drop them in the chat.
Thanks, Elliot.
Perfect. Thank you. Appreciate it. Appreciate the opportunity to speak to everyone today. Hopefully everyone can hear me. I'll keep my camera off for the moment, but I'm happy to turn the camera on, I guess, when we reserve bandwidth, when we have questions and whatnot, so we can talk one-on-one. That said, of course, we spent a little time looking at how we can improve and control and uniform, create a more uniform bioreactor process. What I'd like to talk to you a little bit about here is, I guess a good problem to have for many in this industry is what happens when we go live and at scale and we start to plug into the existing supply chains that exist for food currently, and what are some best practices that we can take away to start that process now? How can they actually complement and help you on your journey towards both regulatory approval, regulatory compliance, and quite frankly, good public relations? See if we can get in there. So first of all, just real quick, my background working for a company called InflexionPoint. I've been working there for 30 years plus now. InflexionPoint is a company that delivers solutions in the space of manufacturing automation. We do also manufacturing information systems. And we do this specifically for life science companies, food and beverage. We have a slice of cultured meat customers that we work with right now who are working very hard to scale up out of laboratory environments. And we do that in part through a bioreactor control system that we actually own and develop as a product. And we also work quite a bit with critical infrastructure. The main theme across all those different industries is regulation and regulatory compliance in one form or another, whether that be, again, to the FDA or the USDA in the United States, or European Regulatory Commission, or even, quite frankly, in the realm of cybersecurity. So regulation is all around us with an industry, and in many different facets. And as businesses, we need to manage that risk. Also do a lot of other public speaking. I work with an organization called the International Society of Automation, and have led that organization through in various positions in their standards and practice development. And I also work with an organization called MESA, which is the Manufacturing Enterprise Solutions Association. So they are a consortium of end users and practitioners and delivery companies that center around smart manufacturing, digital transformation. And quite frankly, I have a podcast as well, if you're interested, which talks about the application of analytics in the realm of digital transformation. So you can check that out as well. Well, always good to start off with a quote. This is just one of many regulatory bodies we'll talk about that are involved in of course, the cultured meat space, which makes this even more challenging for those of us that are practitioners in this space. But the general gist of the presentation or idea here is that while the regulatory bodies may set the framework for what is required in order to meet that threshold of standard in a particular market, it is ultimately up to the manufacturer to make sure that they can meet those requirements. And quite frankly, it's good business to be able to determine and ensure that, through the application of practice of delivering food that meets regulatory requirements, that we are also delivering food that is safe and that consumers can enjoy and know that is a responsible, well-built product. Talk a little bit, just as a high level in the topics for today. I want to talk a little bit about the challenge in alternative protein production, regulatory requirements. We'll talk a little bit about some horror stories, I guess we'll put it that way, in the life sciences to stay away from. Then we'll talk about practical strategies and enabling technologies. So first off, I don't need to tell anyone this, but obviously the current food supply chain is very complex. This is a USDA slide and picture. The green highlighted areas, what's interesting about cultured meat is that, as you're aware, at least in the US, what has developed out of the consensus and the regulatory bodies is that it's kind of a hybrid, just like the products themselves, in that the FDA feels that they have the expertise to regulate the manufacturing of cell culture because they're coming out of the biotech space, where that technology, in fact, is coming from. Much of that technology, equipment, suppliers, knowledge base, etc. But then on the back end, when it comes to actually taking that product in and using that product in various forms and delivering it out to the consumers, it becomes the USDA's responsibility. And that handoff is still somewhat of a work in progress, but there have been some sort of decrees and regulatory frameworks that have been built out, some statements that have been made by both organizations as to how they would handle this as organizations begin to really be able to build their product out to supply chain level manufacturing. Another interesting thing that I think, when you look at the complexity of this supply chain, we have something else at our disposal as an industry that perhaps the existing food industry did not have in the many years that they've had to develop track and trace capabilities through this nebulous supply chain. And that is, we have technology. We have digital transformation technologies. 75% of investments from food and beverage are in track and trace type technologies, applying technologies to improve visibility into the supply chain in various ways and forms in order to ensure the quality and the delivery of the product that they're making. So again, I don't think I need to educate too much on to this audience yet, but I will just to level set, what is unique about the cultured meat industry and how it will plug into the supply chain, of course, is that we are collecting then, of course, and culturing cells to develop up the product. But eventually, once that product is made in some form or fashion, whether it involves just cell replication, or if it involves a more complex scaffolding and assembly of cells into a more refined product that more represents, let's say, what you would get out of an actual animal as a product. Either way, it ultimately goes into the supply chain, and it goes into the supply chain, and it's made into as something that's a mixture, essentially, that may combine both plant-based product and meat product, or it may, as I pointed out, be a more complex protein type product that looks and has the texture of and the flavor of, and it can be cooked and prepared the way even a traditional muscular meat would be prepared. Of course, the challenges here are tracking cell lines and production processes, which is something comes right out of biotech and life sciences, and then ultimately tracking the supply of stock, further processing, and ultimately, in some cases, for some of cultured meat manufacturers, they've decided to go into direct branding, which I think is kind of an interesting choice. That they feel that they can reach their consumer faster and make an impact faster by being able to take their product to market in various ways, direct to the market. But eventually, at scale, and that's where I think we all want to go to. We want to be able to deliver product through existing supply chain channels. And that involves then working directly with the existing cold chain and understanding and leveraging existing networks. Excuse me. So how do we fit into these existing supply chains? We definitely want to take advantage of existing infrastructure, take advantage and understand existing logistics practices. We definitely want to leverage existing distribution networks. So to contrast, like I said, go direct to market, go direct to your consumer. It may be great from an initial splash, from a marketing perspective, from the perspective of getting the word out about what this product is and that it's safe, and developing a groundswell of enthusiasm around what's being done in culture meat. The reality is, at the end of the day, to scale, we were going to want to plug into an existing distribution network. So it's important for us to understand how we're going to do that. What are the regulatory requirements for traceability? What are those minimum requirements? How do we plug in? How do we interpret those? And having a statement about what we, as an industry, as a manufacturer, believe those requirements are and how we're going to apply to them. Because we have as much say right now as this regulatory framework is still forming as to what we believe we need to comply with. And we have an opportunity to have a dialogue with the regulatory agencies about that. And ultimately, like I said, we're really driving towards enhanced consumer confidence. The ability to plug into existing networks, ability to use streamlined logistics and gain operational efficiency really allows us to have be a product that is no different than any other product. And that may sound like, contrary to what we're trying to do. We don't want to be a differentiator. We don't want to be something unique. The flip side of that is that, well, people have a lot of confidence in the food that they eat, and they are very disturbed when they find that the food that they are eating is not safe to some extent. We want to clamp onto that. We want to grab onto where that confidence has already been built through the regulatory framework, through the existing supply chains, and we want to be to deliver as much confidence to the consumer that this product is being delivered to you in a safe way, no different than any other product that you're used to consuming in the past. Again, just to review a little bit of the regulatory complexities here, in particular in the U.S. where I'm based in North America. There has been a tete-a-tete between the U.S.'s federal FDA and our USDA about where the transition point would be and who has the best expertise in order to be able to evaluate, regulate, and keep the culture meat industry in check, if you will. That all said, it's interesting when you go out to even the FDA, when it talks about food processing, and of course the FDA regulates food into the consumer marketplace, but only certain types of food, meat not being one of them. That's USDA's purview. They have introduced some interesting concepts which are very important for track and trace. The idea is CTE, or critical tracking events, and KDE, or key data elements. And again, the idea is here, as as you are progressing through the manufacturing process, as you're progressing into the fabrication process and the differentiation process to now make what is basically a collection of cells now of a certain quality and genealogy, all the way to a product now, that you need to define along the way what these critical data points are that you're going to collect. And those critical data points need to be then in turn tied back to some important process attribute, some quality attribute that is a demonstratable attribute. You can validate and run a protocol testing against that to demonstrate that by keeping this particular data point in a particular range, in a particular check, that you are delivering a quality product out of this particular point in the process. That adds complexity, but also simplifies some things, because it gives us a baseline for understanding what it is that the FDA will look for when they want to understand what are you tracking throughout your process. What are these, again, key data elements, critical tracking events that you want to advertise that are important for your process, and what is the science behind them? And then anything else that needs to happen that is variable to the process, for example, using different types of equipment or different cell lines potentially that can yield the same CTEs and KDEs, well, that would be an acceptable way to approach both the regulatory requirements and the supply, and quite frankly, good track and trace practice to be able to know that we're delivering quality product. Contrast to what happens in the USDA and where that gets handed off within the market in particular. So let's take a step back and talk about regulation a little bit. I like this story because I've worked in life sciences for a very long time as well. And as I mentioned, we have a bioreactor product and bioreactor control system. So we've done a lot of work in biotech and cell and gene therapy, et cetera, and in culture meat. But we also work throughout the life sciences supply chain. And in one of the set of projects that we've been working on over the years, this concept of e-pedigree or supply chain security, which really kind of gained its genesis in 2006 through some regulations and groundswell of thought that came out of California initially, and then was grabbed onto by the United States, and they came up with a set of regulations. Again, the EU followed suit and gained global alignment in 2019 on how they were going to essentially track the unique, not necessarily uniqueness, but also the efficacy of a product out into the supply chain. They're in implementing something as complex as trying to manage the supply chain. So I'm producing a medical product. Very often the use case is a consumable, a tablet of some sort. And I need to know that that tablet being manufactured in my plant ultimately is serialized in such a way that I can track that out through the supply chain, ultimately to the consumer, because the consumer wants to know that they've gotten a true product, not an adulterated product, not a counterfeit product, a product that is essentially the same product that was manufactured. A lot of different complexities, as you can see here, being able to track the unit package out to the carton, to the master case, to the pallet, and numbering conventions and ways of approaching this that have been written into the requirements, into the regulations, et cetera. And many organizations spend a lot of money trying to comply with this. There's been many extensions that have been added to this regulatory set of requirements, and many exceptions now that have been added to that. Because the reality is, once you get out into the supply chain, it becomes difficult, right? Even for something that seems to be as unitary as a container of tablets of a medication, that container comes into, when it goes to a pharmacy, it goes into a certain size container. But ultimately, when I have a prescription filled, that container may be dumped into a container that is relabeled by the pharmacy for my consumption now. And that genealogy gets broken because it gets combined with a second container that came from a different lot, from the same manufacturer, same potential potency, same potential quality, but the idea that we can track genealogy and this serialization all the way through to the consumer is extremely complex. As I mentioned, the results here to date, significant costs and complexity, still many blind spots in the supply chain. The standards have emerged to some extent. If you're interested in looking at this, OpenSCS, which is a technology standard, which talks about implementing technologies in packaging serialization, is a good place to start to understand how you can implement solutions like this. But the reality is, even the best intentions of regulations, we need to have a conversation with the regulatory bodies to make them understand how they can best help regulate us so we can both together deliver quality product and track that product through the supply chain. Culture meat will be no different. You'll be selling the product ultimately to folks that will change that product, add that product to other parts of a supply chain, other products that they bring into the house, ultimately to generate something new that ultimately goes to the consumer. And so what's practical to implement for traceability? I would say first, look at the supply chain, the strategy from a business perspective that you're bringing to the table. Many of you, may be working at the laboratory, the scale up level now, but from a business perspective, how do you want to go to market? How do you see your product being used in various ways? And who are the partners you're going to be working with in order to deliver to that market? And then start to take a holistic approach and look back, of course, at all the different control points within traceability and what are the key variables and data that you're going to collect. Look for technologies that you can implement, okay, that are going to take that holistic approach. In other words, don't look for a specific technology in the scale up and in the bioreactor chambers, and then look for a different technology when you're fabricating, and look for a different technology again when you're out and selling that product into the supply chain. Are there technologies and techniques, and there are, where you can implement data collection and integration across a wide variety of different systems that you'll encounter in order to, again, normalize that data and generate the data that you need in order to support your regulatory compliance and your traceability, because traceability data is not just useful for regulatory compliance, but it's useful for you as an organization as well, to know that you're delivering quality product and you have control over that product as deep into the supply chain as practical. Again, looking back to the existing food industry, not just KDE identification, which is something that, again, you can look back at FDA examples. There's some great use cases out there on how to apply and identify and advertise the key data elements that you're going to track for or to track and trace. But also, quite frankly, just HACCP testing is another concept that's very important, already ingrained within the food industry. Process monitoring, data collection, you cannot collect enough data, even if that data is not supporting KDEs directly. And product release testing is very important. All these different areas here, if you look through the supply chain, they all generate data. They all generate different types of data. It may be data you own, it may be data that you don't own, but you are responsible for having some sort of way of pulling all that data together to create a full traceable view of what your product is as it goes from an initial cell line all the way through to ultimately the consumer's mouth. So again, I like to think about this from a digital transformation perspective, right? That if we have these different areas of the supply chain, research, cell growth, manufacturing at scale, cold chain logistics, ultimately the consumer, that each of these has sources of data. And rather than trying to build systems specifically for each of these areas, what we really want to do is integrate at a higher level to the systems that are there, okay? And implement the ability to grab data, extract, transform, load, that's what ETL stands for, integrate that data and pull that data into a unified platform. And then from that platform, be able to generate the data that we need to support the full traceable view. That's the key, right? And by doing this and approaching it from this perspective, essentially, we can now generate a view that today I may not be fully integrated, fully in production, fully scaled out yet. But if I have this holistic view of how I'm going to apply technology, I'm ready to apply this same technology and technique as I expand, as I bring on new partners, as I look to grow my business, and maybe I don't do everything in-house. Maybe I have to work with other partners, other logistics providers, etc. And ultimately, I can still bring that data in and have that uniform view. Couple of things that are key. Obviously, we talk about the data strategy, which is something that I'm always interested in talking to people about, is how to make the data, how to normalize the data, make it more holistic, so that we can pull that data in across different disparate sources, but have a unified view of that data, which is critically important for track and trace. But there are some technologies that we want to definitely look at that are very important, that are already being used in the food industry, and implement and continue to implement those technologies. Things that seem somewhat maybe either ubiquitous or boring in the world of digital transformation, but they are critically important because they're proven, and we want to use proven technologies because that can simplify our ability to maintain the technologies and get to market faster and get reliable data from these technologies. And again, those would be barcodes, RFID, things of that nature that are already being used out there in the industry. Real quick, just to review some of those technologies, tracking technologies such as RFID or Zigbee or LoRaWAN. These are technologies that are proven. We can use those to track product through the supply chain, essentially. So again, you have a product that defies being labeled by itself. It moves from container to container, vessel to vessel. We want to be able to track those in a passive way, but make sure that we are tracking those products accurately. So look for passive technologies that can be applied, and proven technologies. Again, IoT is our friend here. We mature as an industry in a world where we have access to these smart manufacturing technologies. So looking at smart sensors, mobility, and continuous monitoring through, again, passive temperature sensors and environmental sensors, et cetera, and applying unified data models across these different technologies. Ultimately, I want to create a view of traceability that looks at my raw materials and my transformation through the various processes and steps and see and be able to track back from finished product deep into the supply chain that I'm currently involved in, all the way back to the original manufacturing. What vessel did it come from? What day was it manufactured? What cell line was used, etc. And I can need to understand then the cause and effect and have reactivity to it. And again, I'm driving customer experience. These are investments that are important to make, not just from a regulatory perspective, but also because, quite frankly, our consumers would be very happy to know that we can do this, right? And be responsive. Very quickly, some technologies that I think that they are available today. I would be avoiding them right now. Again, serialization is one of them. It's complicated. Again, getting into the supply chain, deeper into the supply chain. When people talk about serialization, I think it becomes very complex. I'd rather have a higher level view of data and again, transition points of a product and what kind of key data I need to collect at those transition points in order to keep track of that data, rather than know specifically what container was used at a particular point in time. And then blockchain is another one. There's actually some good work that's going on in blockchain. I have some hyperlinks down here, but the reality is it is a technology. It needs a lot more work from a regulatory perspective to understand how the technology will be applied. That's what I would say about blockchain, which is most in need right now. It's not so much that it doesn't work as a technology, but that blockchain talks about this idea that, well, it's going to be out there and everybody can plug into it. It's going to be open and therefore verifiable. Every blockchain implementation, even in the cryptocurrency world, requires some sort of body that is overseeing it. And so those two contrasts right now require some maturity in how blockchain will be implemented before we can effectively use it to implement it in track and trace, I think, in the food industry. So key takeaways here: you are responsible for the quality and the data is the key. Look to the technology today that is available. Cloud-based technologies don't necessarily need to implement stick-built solutions in each of these different vertical areas that you're working in. Rather, look to how am I going to collect that data, the right data, the data that I need, and put that into a cohesive centralized data store so that I have continually a way of getting that data in one view, one picture. Take advantage of digital sensors, edge devices, telemetry, the things that are available to us now. Don't neglect security in your configuration for your network. We don't want data to be compromised. Essentially, we want our data to be accurate. We want it to be reliable, etc. There are lots of good ontologies that I can talk about offline. Happy to talk about on how that data can be then mapped and then represented in order to give you that unified data view. Again, analysis, awareness, visibility, that's the key to traceability. And that is it. I could be contacted along with my partners here on the line with IXP anytime. Again, some sources of information, but I'm happy to stop here and take some questions.
Awesome. Thanks, Chris. That was really interesting, especially for me. I don't spend too much time myself thinking about these things, so it was very educational. And I think sort of timely. I know there was a food safety E. coli outbreak at McDonald's just like yesterday, and I think or 2 days ago, and I think they were able to narrow it down very quickly to the onions as the onions from Idaho or something.
Right.
And it is basically just because McDonald's has such good supply chain traceability that they were able to sort of identify the
Those
causative factor
success
here.
stories are wonderful because we should be asking ourselves in an industry when we see that, how did they do that? How did they do that? And what are best practices they're following? Who should I be reading over there at McDonald's or whomever the organization is? Chipotle is kind of another one too. They had an outbreak a couple of years ago and they got dinged a little bit because of course their brand is all about freshness and whatnot. And they had a new CEO come in and really change the culture and made a point of publicly discussing the failings, but then rectifying the ship. And they have a nice story to tell too about how they changed things and matured as an organization as well. There's lots of good stories out there, lots of concerning stories, but also lots of good stories. I think that we're at a point now in the food industry where we have a lot of mature technologies and best practices, and I encourage people to when they look at both the regulatory requirements as they're dipping into a higher scale manufacturing, and also the need to again brand themselves as a safe product to the consumer. You're not going this alone. There's lots of good stuff and lots of good best practice out there to learn from in order to pick the right technologies and the right approach to scale. And a lot of us are not really at the point in this culture meat industry where we're ready to scale full bore, although many of us are at that point. We're getting close. And so this is the time to start thinking about it. And what's the right level of investment? What do I need to do in order to set myself up now and in the future for growth?
Makes sense. Great. Well, I think we're right at the top of the hour. So thank you both for presenting. I know it's a sort of light live audience today, but we'll get these presentations out to our wider community, and hopefully people will take a look and reach out to you. But yeah, both really informative and I think useful. So thanks so much.
Thank you for the opportunity. Thanks for everyone who attended. Absolutely.
See folks next month. Take care.